Okay, so in my part, I'm going to share the purification part of third generation sequencing. And my presentation divided into 3 parts. First, I will explain the request of third generation sequencing about the purification part. And second, we will discuss a little bit about the problem of current purification. Then we will go to the solution of novel purification solution for third generation sequencing. Okay, so let's jump into our first part, request for third generation sequencing. Okay, first of all, this is an evolution of sequencer. In this picture, we can see in the past 2 decades, our sequencer evolved from first to next. Even right now, we can reach to third generation sequencing. And in third generation sequencer, there are 2 major players. One is Oxford Nanopore. Another one is Pacific Bioscience. And I believe lots of persons attend this webinar already well know of the principle of the third generation sequencer, but let me introduce briefly. For nanopore, actually it's the protein nanopore is embedded in a synthetic membrane. Okay, and for Pacific Bio, actually it's a DNA polymerase in the bottom of the well. And for Oxford Nanopore, it's single molecular direct electrical readout. Similar to Oxford Nanopore, actually Pacific Bio also single molecular, but it's real-time synthesis of the DNA molecular. And for Oxford Nanopore, actually it reads the ionic current disruption as DNA passes through the protein, and it can detect the current difference. But for Pacific Bioscience, actually the fluorescent signals from DNA synthesis can be detected. So therefore, it can do the sequencing. Okay, so since the principle of these 2 kinds of sequencer actually is well known, right now we move to the request of DNA because actually they got special request of DNA quality. First of all is the DNA integrity about the fragment length. Actually, both of them need to better to have high molecular weight DNA, ideally 20 kilobase, and better will be 30 to 50 kilobase. And longer fragment produce longer reads and better assemblies. Second, about the DNA, it's better to minimal degradation because it can be read longer. And to get the minimum degradation, it's very important to avoid free throw cycles and sharing and avoid harsh extraction. Third is about the purity. For the purity for the absorbance of 260 and 280, it's better to have the ratio better to have 1.8 to 2.0. And this ratio indicates minimal protein contamination. And both absorbance of 260 to 230 better to over 2.0 because it will indicate minimal salt and phenol contaminant. And better to know inhibitors inside, like in detergent, polysaccharide, or ethanol, because this will inhibit polymerase or nanopore function. So have a brief conclusion of this requirement is integrity and purity is super important. The DNA integrity and purity is very, very important for third generation sequencer. Okay, so let's move to the second part of my presentation about the problems of current purification method. First of all, let's see the factors will influence the DNA integrity. There are so many factors will influence, especially during the before purification and during purification, such as vigorous vortex definitely will destroy and share the DNA into smaller fragments. Also, repeated freeze and throw, and also high temperature incubation and pipating through narrow tips. Unfortunately, actually, those factors are avoidable because we can skip or avoid such step to avoid this happen. Other factors like shearing stress. Here is an example. Okay. This is the agitating mixed step of magnetic beads-based purification system. And actually, this kind of agitating will, it is during mixing, beads keep shearing stress to cut DNA into smaller fragments. So it's kind of shearing stress will degrade DNA into smaller fragments. Another type of factor to influence integrity is we call centrifugal force. And here is a video to show it. And it actually comes from centrifugation step of column-based purification method. And during centrifugation, centrifuge will provide very strong G-force to share DNA into smaller fragments like this. Especially the DNA path through the membrane, it will destroy. Unfortunately, this kind of factors which will degrade DNA usually is inevitable because usually this kind of shearing stress or centrifugal force usually belong to the major step of purification. So later we will have some example how to solve it. And about the factors will influence the DNA purity. There are so many impurities like salt, alcohol, phenol, protein, polysaccharide, or fragmented DNA or RNA, or salt or alcohol. Actually, for salt like allotropic salt or alcohol like ethanol or isopropanol, usually this kind of impurity is from reagent, from lysis buffer or wash buffer. And the way to eliminate, to remove it, actually depends on very efficient wash steps. And phenol also an impurity. And the function of phenol usually used for protein removal, but it's not very good for environment. To eliminate it, it's very simple. Just avoid to use it. And for protein and polysaccharide, actually it's indigenous from the sample, from the cell or tissue. Proteins like histone, cellular protein or membrane protein, and polysaccharide, especially rich in plant tissue or some bacteria. And the way to get rid of this kind of impurity is to treat with proper buffer and also efficient wash step. And finally, about the fragmented DNA and RNA, we know during pre-treatment or extraction, sometimes DNA degraded into smaller fragments by too vigorous pre-treatment. And also RNA will co-purify with DNA. In this case, we need additionally step to eliminate like fragmented DNA or RNA. So after all, about the purity, we can get a small summary that a powerful wash step is very, very important for get higher purity. Then we move to solutions about purification solution for third generation sequencing. And in this part, first of all, I need to introduce eye catcher system because eye catcher system, we have CAPD cap technology inside, which means coated membrane, anti-gravity, porous membrane, and positive pressure. actually, this system was developed for liquid biopsy. But why I mentioned about it, later I will explain a little bit about it. At the very beginning, while we developed iCatcher, actually it's designed for liquid biopsy. We did some market survey because for liquid biopsy, actually we need handle large sample volume. And usually, while the sample volume is larger, there are more impurities like protein, like salt, like ethanol, or other impurity inside. So therefore, we need a system which can purify, which can get higher purity and larger sample volume for liquid biopsy. iCatcher was designed in this kind of situation, which can handle 4 milliliter sample. And it's a membrane-based system, which we designed to capture more cfDNA and cfRNA. And also, we built in a kind of wash method. We call it wash-away wash method, because it can get cleaner of cfDNA and cfRNA, because it's very, very rare. Need to be very high purity. And here, let me show the video of iCatcher first, and then we will tell you the story why iCatcher will able to purify large fragment DNA. Okay. So in iCatcher, we have built-in touchscreen, and we can choose different kit. Just simply type in the number. And in iCatcher system, we can handle up to 4 milliliter sample, which means 12 milliliter lysate. Okay, and here is the binding step of iCatcher, which is a very critical step. I will explain later. And after binding, we do several times of wash on the iCatcher, and it's a wash-away method to drain the wash buffer through the membrane and dispense it out. This is third time of wash. Then we use heating to evaporate ethanol to remove inhibitors. Finally, we can elute in only 30 to 200 microliter elute and get very pure DNA inside of the elute. So for iCatcher, actually it was designed for cfDNA purification. Therefore, it's a membrane-based wash-away wash method that can handle up to 4,000 microliter samples. And here comes the story about why iCatcher is a good solution for large fragment DNA purification. Actually, it's a beautiful accident. Why I call it a beautiful accident? Because someday we got a data from our customer side, and this data this data actually from a customer who do the quality control of genomic DNA. But this customer actually don't perform third generation sequencing. They only perform next generation sequencing. But they, in their protocol, they need to qualify the fragment size. Then the data shows two results. First one is the result purified by catching manual kit as positive control. The result is over here. The GQ1 is not very good. Another one is the result comes from eye catcher automated kit, which is over here. There are 14 different samples. And for DNA purified by eye catcher, actually, we can notice the integrity of this DNA purified by eye catcher actually can get very high GQ1 result in average in 8.7. And the average fragment size can reach to 42 kilobase pair. But for manual, only GQ1 number only 4.3 and fragment size only around 11 kilobase. We are really curious about the data because actually for eye catcher and the catcher manual kit, we use same membrane column, we use same reagent. The only difference between these two methods is the way to complete the whole workflow. For manual kits, we use centrifugation because it's typical column-based purification kit, so it's based on centrifugation. For eye catcher, we use positive pressure to let the lysate on the membrane to pass through the membrane. So this beautiful accident brings us a very important information. Actually, for eye catcher, it's unlocked another secret technique. Actually, this data unlocked another secret technique for eye catcher, which shows maybe based on this principle, eye catcher is able to purify larger fragments, even it's based on membrane. Okay, so let's go deep into the data. Here is the manual spin column, the capillary electrophoresis data of the manual positive control. We can see here is the genomic DNA. So the size is not very big. The fragment size is not very big. But based on eye catcher, based on positive pressure of binding and washing step, we can reach to very high and larger fragment. And it can reach to 39.3, 48.5, or even to 50.3. Some of them even bigger, almost reach to 57, something like this. And in our internal, we also curious about if we can repeat it, and we did lots of tests on our eye catcher. We also purify by eye catcher and run on our QSEP capillary electrophoresis system, and found based on eye catcher, yes, we can get higher integrity, like 30 to 40 or even to 50 kilobase in fragment size. And not only by our internal, one of our customers, they use our eye catcher to parallel compare with QBRAM manual kit. Here, I put it as a QBRAM because we can see the complete name of this company. And it parallelly side by side to compare. And go into the nanopore sequencing. We can see the parallel comparison. Eye catcher purified genomic DNA is in orange color. The read length is in orange color, and the QBRAM is in blue color. And the read length and the read amount of eye catcher is higher than QBRAM. And it's obviously difference between eye catcher and QBRAM by sequencing by nanopore. And besides, not only large fragment of genomic DNA, we also did a parallel comparison, a parallel demo at customer side. And we also found in the FFPE sample, here, the black line, the black curve is the DNA purified, FFP DNA purified by QBRAM magnetic bead system. And the red curve is the FFP DNA purified by eye catcher, and we can see eye catcher get higher integrity. Here is the DD200 line, means fragment larger than 200 base pair, and the DD200 ratio of eye catcher is much higher than pure. So this shows the difference of the two methods. And so here I will illustrate how eye catcher can get higher integrity. Here we already saw it for magnetic bead system. Because of agitating, it will cause shearing force and will cut DNA into smaller fragments. For manual column-based extraction method, it will cause DNA will degrade it by centrifugal force, so cut it into smaller fragments. And for eye catcher, actually we already saw it. Let me play the video. During the binding step, actually we use positive pressure to let the lysate pass through the membrane, and we do the bind and wash steps gently. So it's minimize the destroy of DNA and also create higher recovery rate because DNA RNA have higher possibility to bind on the membrane. So its cost, we can get higher, we think it's the major cost. We can get higher integrity of genomic DNA. Okay, so remember in the first section, we mentioned about another important factor is the purity. Not only integrity, actually eye catcher can get higher purity. And how can eye catcher can get higher purity? Here is the reason. Okay, this is magnetic bead-based purification method. Let's see how it works during wash step. Okay, so during wash step for magnetic bead system, it's just like soap in the bathtub. Beads are released in different ways, and just like take a bath to remove the impurity, like in dilution method. But for eye catcher, we are different. We use a wash method. Here is the video shows the difference. Okay, we use wash method to just like take a shower to wash inhibitors away. Okay, so here is the data also from the built for accident customers. Here is the genomic DNA purified by eye catcher and check the integrity. Not only check the integrity, they also check the ratio of absorbance 260 to 280. And all of these 14 samples reach 1.8 to 2.0. And also for absorbance 260 to 230, it's all over 2.0. And for a third generation sequencer, usually we also care about the qubit to nanopore ratio. And all of this result is it's in the range of 0.8 to 1.0. So it's a very good purity. So we believe not only high integrity, but also high purity can reach better result for third generation sequencing. Okay, so here is, so for eye catcher, actually it's formed for liquid biopsy, but it's a great automation purification solution for third generation sequencing because we can get higher integrity and higher purity based on the special method. Here is the internal view of our eye catcher system. And for eye catcher, we have a kit which pre-fill the reagent in cartridge, and we have a special syringe which can handle large volume, and all the consumables are inside of our kit. And it's so easy to set up the work table, just in 5 steps, in 10 minutes. Put the syringe, put the cartridge, and place column, place adenole, place sample. then can click the start to run the system. And we have many different, actually we not only have kit for DNA, we also have kit for RNA, DNA, and also for CNA, which means circulating cell-free DNA. And we got patents of Japan, China, Taiwan, Europe, and USA. And we also passed the certificate of safety and EMC, and we got ISO 13485, and also some licensing for medical device in Taiwan. And iCatcher, of course, also got the FDA listed. Okay, so here is my presentation, and we think iCatcher provides a simplified your purification of third generation sequencing. Okay, I'm Alex Lee, and here is my email address. But if you need it, you can just leave some message. I'll give it to you. It's a little bit messy here. Thank you.
Thank you, Alex, for the great presentation and for sharing those detailed insights. Next, we're very happy to have a special guest, Thomas. Thomas, the floor is yours now.
Hello. Let me connect my PowerPoint. Okay, can everyone see my PowerPoint and hear me clearly?
Yes.
Okay, good. So first of all, thank you for the introduction and invitation to provide a user feedback session in the project, in the context of the project I'm working on at this moment. Welcome everyone, I'm Thomas Verboven, and I'm a research technician at the St. Antonius Hospital in the Netherlands, and I've been working on this project and with the iCatcher for the past 9 months from now. So
Thomas, I think
yeah.
the PowerPoint hasn't been full screen.
Sure. Now yes, I guess. So what is the project? Antibiotic resistance is one of the major emerging problems worldwide at this moment. And as the World Health Organization reported, there's a significant increase in multi-drug resistant pathogens reported in the past few years, and they reported that it will increase even more. So therefore, it is really important to understand transmission events of these multi-drug resistant organisms and study the epidemiology. Therefore, in the study, we aim to type strains and do cluster detection and identify transmission patterns. And we did that by sequencing a lot of multi-drug resistant pathogens like Klebsiella pneumonia, Escherichia coli, and Enterobacter complex from the region Utrecht, and try to detect transmission patterns and clusters. But what do we need for that? We need high quality DNA in order to get sufficient sequence results. So which workflow did I use? We work with 3 other collaborating hospitals. So first of all, I collect all the samples from the hospitals and from the region I'm working in. After sample collection, I did the iCatcher DNA extraction. After the extraction, we immediately went to whole genome sequence the strains and the isolates by using Oxford Nanopore. And after that, we do the data analysis. Then I want to tell a bit more about the optimization of the DNA extraction I did. I had 3 steps within this optimization. The first method was the standard protocol, which is included in the eye catcher kit, in which we calculated that we need 100 microliter microbial start culture. That didn't work that good, which I'm going to show you later. So we went on with method 2, which is our standard, which is again the standard protocol with the same initial start microbial culture, and we added the bead purification afterwards to try to purify our elutes a little bit more. But the downside of this this step is that it takes a lot of time. So therefore, I went on with method 3. Also the standard protocol, we increased the start culture to 300 microliters, and we did an extra RNase A addition step, which is much quicker than the bead purification, and is included as an optional step in the eye catcher protocol. So I'm going to show you some results from our optimization of the DNA isolation. First of all, it is really important to generate enough data within your sequence run, otherwise you can't align your whole genomes and you don't have reliable data. So therefore, we wanted a sequence depth of at least 40 for every strain, and based on your sequence depth, we can calculate our threshold, so the amount of data we want to reach in order to be able to align most of the sequence strains. We did that by the sequence depth 40 multiplied by the amount of strains we were sequencing with one flow cell, and multiply that by the genome size, and that came at roughly 15 gigabases of data. So that was actually our aim to reach per sequence run. So as you can see here in gray, which was a normal eye catcher isolation method, we did not reach our threshold of 15 gigabases of data, which resulted in the fact that we were only able to align just a few strains per sequence run, which makes the workflow really expensive if you can only align a few strains. That's why I went on with method 2, the same protocol, 100 microliter start culture, but with the added bead purification afterwards, and that significantly increased the amount of data we got from our sequence runs, but still did not reach a threshold of 15 gigabases we wanted to reach. So we were still not able to align all the strains within our sequence run, and combined with the fact that the bead purification takes a lot of time, we decided to further optimize the protocol. So that's where we implemented method 3, in which we removed the bead purification and did the additional RNase A step, which results in the removal of lots of RNA, so lots of small fragments. And we ran sequence runs with this, and we actually this actually resulted in lots higher data output, and far above the 15 gigabases of data we want to reach. So here we actually optimized our protocol so good that we were able to align a lot of the strains within our sequence run. And this is supported by the graph that we see here. And at the left axis, we see the number of available pores within our sequence run, within our flow cell. And as Alex told, the pores are actually the sequence mechanisms within your flow cell. And at the bottom axis, we see the time. And as we expected, when we have more pores over a longer period of time, we are able to generate a lot more data. So here again, we have method 1, which shows a very quick decline in available pores in the few first hours, which is probably due to impurities or maybe a lot of small fragments DNA. And after the optimization, we see method 2 again in blue and method 3 in purple, which both show similar results. We see a stable decrease in the available pores over time in the first 60 hours of sequencing. The flow cells ran much longer and generated more data. And since we have more data, the alignment quality and sequence quality improved a lot by using the method 3. So what did I do so far within the project? Of course, we ended up using method 3 with the RNase-A addition. And in total now we sequence around 500 isolates, of which 220 Klebsiella pneumonias, 259 Escherichia colis, and 21 Enterobacter cloaces, with for each species an average DNA concentration of roughly 20 for E. coli and Enterobacter complex, and a lot higher for Klebsiella, around 30, which are actually some good concentrations to start our sequence run with. The average sequence depth we're reaching at this moment using our optimized method is also a lot higher than we want to reach. So that's also a really good thing for Klebsiella and E. coli. We reach almost 100, and for Enterobacter complex, we actually reach far above 100. And that's also what we see here. Of course, we have some strains which don't reach the average sequence depth, but we can repeat them in the next run. And sometimes we even reach sequence depths of above 150, which is really a lot. And the average M50, which is really important for sequencing because it indicates your initial fragment lengths when you're going to sequence, is 6.38 kilobases, which also increased over time during the optimization of the methods. We started with method 1 with an average M50 of roughly 4.0, and at method 3 we're stable at 6 and above. And one of the most important things is that we have a repeat rate of only 6% of the strains within our sequence runs. At this moment we run 36 strains per flow cell, and which means that we only have to repeat like 2 strains within every run. So that's really good. In long-read sequencing, sample integrity is everything, and that starts with purification. And I think I can prove that by these results based on the optimization of the DNA isolation. We are very happy with the results and also with the extraction quality of the eye catcher, which is, in my opinion, very easy to use. We have minimal handle time, and most important, we see consistent isolation and sequence results. Thank you for listening, and feel free to reach out if you have any questions after the webinar.
Thank you so much, Thomas, for sharing your practical experience and real-world results. That was really insightful. And to wrap up today's session, we will now move on to our final speaker, Alice, who will highlight how QCEP systems simplify DNA qualification and analysis. Alice, please go ahead.
Okay. Hello, everyone, and thank you so much for joining today's webinar. My name is Alice. I'm the BD Director of BiOptic. And today we will focus on one of the most critical factors in the third-generation sequencing, that is the quality control. So yeah, let's start it. Okay, so what you can expect from this session? The presentation will take about 30 minutes, and firstly, I will start with a short introduction, and I will briefly walk you through that what the third-generation sequencing is and why the DNA control plays such a critical role in this workflow. And secondly, we will walk through the third-generation sequencing workflow step by step and highlight what kind of QC checkpoints are important at each stage. And also, thirdly, that you also will learn about the practical tool that can help you to assess sample quality and get a reliable results. And in the end of this session, I hope you will have a clear understanding of where and how QC impact your workflow, and know how to monitor at each step, and also get an effective QC solution to secure better sequencing results. Yeah. So it refers to a group of DNA sequencing technology that can generate much longer read compared to earlier generation platform. So unlike first and second generation sequencing, that which rely on the PCR amplification and also produce short-term read, the third generation sequencing using the use a single molecular sequencing, and that is no amplification required. And because of this, it can generate long, continuous read and large span complex and repetitive genomic region, and bringing several advantages such as like more accurate, comprehensive genomic assembly, and also better transcriptome analysis, and also we will have more effective structure variation detection. And also because of those advantage, we can use third-generation sequencing in many different fields, from research to clinical and even environmental studies. So let's look at some key applications. In a basic research, the third-generation sequencing help to build complete genomic assembling and help for and study full-length transcript, and also detect the epigenetics modification directly. So it gives scientists a clear and a more accurate view of the gene structure. And for the agriculture, that's used for plant and crop breeding, and also wildlife conservation and microbiome study. And this help improve the productivity and also protect the biodiversity. For the microbiology and also infectious disease is widely applied to virus and bacteria genomic sequencing, and also metagenomic study. So those applications are critical for pathogen surveillance and also outbreak response. And in biotech and the pharma, it support for cell line and the process monitoring and gene therapy development and also nano-drug target discovery. And because the long read provide more accurate insight and for the therapy innovation. Finally, that is the environmental and the public health. It's used for ecosystem monitoring and the biosurveillance, and it helps track change in the microbiome community and detect potential threats early. Okay, so after we finish the small introduction about the third generation sequencing, and let's move on the workflow of the third generation sequencing. So the process typically starts from the DNA extraction and followed by the quality control. And depending on sample type and experimental goal, so you might include the site selection step before the library preparation. And after that, the library will sequence and then analyzed. And also at each stage, the quality control play a critical role to make sure that downstream results are accurate and also reliable. And after DNA extraction, the first key step is quality control. And as I mentioned earlier, that extraction is the foundation of the entire sequencing workflow. So if the DNA isn't clean or isn't intact, everything downstream will be affected. So here now we mainly look at three important QC points. First one is purity and the quantification and also sizing. So I will go through each of those in more detail in next few slides. The first and the most basic check is the DNA purity. And for both ONT and the PayBio, that's recommend is A260-280 ratio is around 1.8, and 260 to 230 ratio is ideally about 2. And those two ratios can give us quickly indication for the sample quality. The A260 to 280 ratio around 1.8 usually indicate the pure DNA with low volume might suggest protein contamination. And for the 260 and to 230 ratio above 2, that indicates minimum contamination from salt, phenol, and other organic compounds. Yeah. High quality DNA should be free from the salt, protein, and also solvent, which can interfere with enzyme reaction during the library preparation and the sequencing. So we usually using the evaluate the purity by absorbance ratio and using spectrophotometers such as the nano-drug. And the next one is the DNA quantification, which is another key factor. And before constructing the library, it's essential to confirm that the DNA concentration is accurate. And for the Oxford nanopore technology, the recommended amount is 1 microgram gDNA for the ligation sequencing kit, and 400 nanogram for rapid sequencing kit, and 100 to 200 femtomole for amplicon or CDN library. And for PayBio, the recommended input is typically 3 to 5 microgram for high quality genomic DNA, and depend on the specific protocol and specific protocol version and also the recovery rate. And accurate DNA quality qualification is very important because it ensures the balanced poor loading and the good sequencing out the throughput. So, but if the input amount is too low, so you will get this throughput and also lower data yield. And if your input amount is too high, then you will actually clog the pore and waste the sequencing capacity. So that's why we use a reliable method like qubit to measure the real double-strand DNA concentration is more accurate and than just using the spectrophotometer, because that is least affected by the contaminant. Okay, so let's start by looking at one of the most important QC checkpoints. That is the DNA sizing. Yeah, for long-read sequencing, the fragment size of your genomic DNA might determine how good your sequencing will be. So, for example, like in the ORNT, the average fragment size should be ideally about over than 30 kb for the whole genome sequencing. And for PacBio, that should be recommend start from the start GDNA from 40 kb. And this session actually we will have more data to share today, and later we will go through many real examples to illustrate how good or poor quality results look like. And in general, high quality long read leads longer read and higher yield. So on the other hand, if your sample is degraded or shared, that will result in shorter read and also lower throughput. Okay. So now let's start to see some results from here. And here is an example of the genomic DNA result. According to PacBio, the technical guideline that high quality genomic DNA should have at least 7% of the fragment above 10 kb, and 50% of the fragment should be above 30 kb. And the result was analyzed by QSAT platform here, and with the N3 cartridge, and under the 4 kb, and the total separation time is around like 7 to 8 minutes. Yeah. So as you can see that the around 70% of the DNA that is above 10 kb here, and the DQ number above 30 kb is over 5. The DQ number now is 5.9, which means that it's the quality requirement that is so good and the qualified sample for next step. And here is another example analyzed under the same condition. This also still using the QSAT platform with N3 cartridge and the 4 kb. And so you can see more than 70% of the DNA fragment are above 10 kb here. And but the percentage above 30 kb, that's lower than 50%. So you can see the DQ number only 3.1. So even though that's the 10 kb requirement is met, but does not meet the 30 kb part. So it's not. It might be do some other steps to treat these samples. Yeah. Okay. So here we have 3 genomic DNA results from here, and that's all analyzed on our QSAT platform and using N3 cartridge as well. So all the 3 cartridges were iterated using the catch-string kit, and the MD that's mean the manual kit, and the AD means iterated by the eye-catcher system, that is an automatic system. So as you see that all of the 3 samples are very clean and also complete fragment profile. So let's indicate very good iteration and the iteration for quality. And on this slide, I want to share with you the reproducibility of the genomic DNA sizing. So the sample on the left side were extracted manually using catch-string manual kit, and on the right side was extracted by eye-catcher automatic system. So both manual and automatic extraction 3 samples were tested, and each of them were analyzed triplicates. And the CD values are all very low and generally under 3%, so which indicate excellent reproducibility. So actually, one of the common challenges for high molecular weight DNA sizing on some platform that is lower reproducibility. That's mainly due to the DNA conformation problem. So, but in our QSAT platform, overall the reproducibility for large fragment sizing look very good and showing very stable and consistent performance. So this slide demonstrates that how the QSAT platform can be used to assess the integrity of the genomic DNA. The simple one, the red one, this one, show high quality of the genomic DNA profile with long fragment peak. And the average size of this gDNA is about 42 kb, and the DQ number is 9.9, which indicates that's excellent integrity. In contrast, the blue one is a sample 2, and that's is a degraded DNA. So the peak was shifted a much smaller fragment size here, around 5 kb here. So you can see the average size is about 5,000, and the DQ number dropped to 5. With QSAT platform, not only can visualize the degradation pattern directly from the electrophotogram, but we also have a clear indicator that is DQ1 value to support the objective quality assessment. Also, here is an parallel test between QSAT platform with the Fento pulse using the same genomic DNA same genomic DNA sample. The DNA was extracted with iterator system, and on the left side, the QSAT result showing the peak size was about 24 kb, and on the right side, the Fento reports results showing that is 22.6 kb. So as you can see, the result is very close, and which indicate that there is good consistency between the QSAT platform with the Fento pulse system for the large fragment analysis. Okay, so after extraction, we perform QC to check the concentration, purity, and the fragment size. If the DNA QC results show high proportion of a short fragment, it means the sample quality might not be ideal for long-read sequencing. Yeah, so in this situation, user can apply the site selection to remove short fragment and also enrich the long-read molecular and improve N50. So this increase the proportion of long fragment in the library, which improve the efficiency of the power usage, extend the average read length, and that can get more stable and reliable results. And now this is the QC result after site selection. And as you see, the short fragment signal are almost completely removed from here, very clean here. And the main peak show now is concentrated in the long fragment region. So this indicates the site selection with high efficiency and successfully enrich the long DNA molecule. So this kind of profile is what we typically want before library preparation. Yeah. And during library preparation process, the quality control plays a critical role to ensure the sequencing success. So after each major step, such as like sharing, ligation, and also clean up, we check both DNA concentration and the fragment size. So the main goal is to make sure that we didn't lose too much DNA during the cleanup step, and the fragment size still remaining our desired size range, and that's the library yield is sufficient for loading. So, and here I want to highlight the two main QC parameters during the library preparation: the concentration and also fragment size. For the concentration, we're using qubit to make sure the DNA recovery is sufficient after each cleanup step, and especially after ligation. And in the both of the PBio and the ONG that emphasize this checkpoint, since low concentration at this stage can waste an entire the flow cell. Yeah. So for the fragment size, we typically verify the sharing was successful or not. That ensure they have, and also ensure have no unexpected short fragments such like adapter dimer or degraded DNA. So the final check are also confirmed that the library structure is intact and ready for the sequencing. Yeah, so it's another parallel test compared with the QSAP platform with the Frontal pulse using the same library sample. Yeah, so both systems were using to check the fragment distribution after site selection for fragment larger than 10 kb. And as you can see that the QSAP results show the main peak around the 20 kb here, and which is very consistent with the Frontal pulse result. That is also another 20 kb. So this shows that the QSAP give a almost the same sizing result with another platform. Okay. And now this slide I want to show the HiFi library result from one of the customer. The first result that's from PBio, the first result from PBio HiFi library QC result show they have a small portion here for the fragment below than 10 kb, and which match to our QSAP QC result here. That's have 77.1% of the fragment that is below 10 kb. And the bottom of the example, this one is the sequencing read length distribution also show a higher portion of a high proportion of a fragment that's below 10 kb. And this is also aligned with our QSAP results here. And this is overall that QSAP platform give a clear and also reliable indication for the library quality before the sequencing. Okay, so after looking through the workflow and show the QC result, let's talk about why the QSAP platform is such a great tool for high molecular weight DNA analysis. One of the big advantage of the QSAP is how easy to use. So if you have experience to use QSAP platform, you will know that just to set up the whole process only less than 3 minutes. And you just load the cartridge, put the sample, and a few clicks, and no need to do any gel preparation, then you can run directly. And also it's very high flexibility. You can run one sample, two samples, few of the sample to 96 sample in one single run, or for large size sample. And the analysis only take about around less than 10 minutes for large fragment analysis. The final one is QSAP platform provide widely application, not only for DNA, but also for the RNA and also for the protein fragment analysis. So it makes QSAP platform a very efficient and a versatile platform to support users' applications. Yeah. So let me give you a quick overview of our QSAP platform lineup. We offer different models to meet wide range of throughput needs, starting from our small model, that is QSAP 1 and QSAP 1 Plus. And this is one of the smallest CE platform in the world. It's only 5.5 kilogram, and it can support 1 to 8 sample or 1 to 17 sample, depend on the model type. And we also have our most classic model, that is QSAP 100. That is a very popular platform. That it can handle 1 to 96 sample. And we have another version that is QC100 Advanced that not only can support regular DNA RNA analysis, but have very good performance in the protein fragment sizing. So, and also for some customers who need very high throughput, we offer QC400. That is 4-channel system, and basically is 4 times faster than the single channel system like QC1 or QC100. Another big plus for with this that is embedded the PC and also the touch screen. So that is a standalone system and no need to connect with your laptop or computer. Yeah. And as I mentioned previously, that the QSAT platform not only cover for large size fragment analysis, our DNA also can analyze such as NGS QC, multiplex PCR, and also CRISPR, SNP, and RGLP, all kind of DNA fragment analysis, and we also can analyze RNA sample, including the RNA QC. We can give the accurate number, and also we can analyze RNA fragment analysis, and some of the RNA sample concentrations if that is very low, it also can be detected in our system. And for the protein application that can analyze protein profiling, or we also can measure the size of the protein sample, and also can do the antibody purity analysis. And because, as I mentioned, that we can do a wide application. So for DNA analysis, we provide more than 10 types of the DNA cartridge. For example, we do have the standard cartridge that can analyze some like genomic, some NGS sample library QC. And also we do have a high resolution cartridge. The resolution can achieve 1 to 4 base pair. And for the faster cartridge, you can do a quickly screen, and each sample only take at least 90 seconds, then you can get a result. And high sensitivity cartridge, and also for large fragment, we do have the kilo-based cartridge, and we also offer the DNA quantitative cartridge kit for some users if they want to get more accurate concentration result. And we also have RNA cartridge and the high sensitivity RNA cartridge, and also the protein cartridge. So widely application, we have multiple cartridge to support customers' application. And because our system that is based on the electrophoresis technology, so one of the advantages for that is very high resolution. Yeah. So here is one of our size marker that's usually included in our cartridge kit. And here is this result. This later is the plasmid digester sample, and here is a 242 base pair and a 238 base pair and a 242 base pair. That is for base pair differences can be resolved from our platform. And this one is a 100 CT fragment, but in here that they have two fragments, the same size but the sequence different. It also can be resolved from here because DNA conformation difference. And we also mix our PCR product with different size to see the resolution of our platform. And that is PCR product that is mixed with the 102 base pair and the 103 base pair. It can be resolved very clearly. And another one that is 200 base pair and the 201 base pair. That's one base pair differences also can be resolved right here. And one of the feature for the electrophoresis that is high sensitivity. Yeah, so here we using, that is the standard cartridge, but we do a serial dilute, and the minimum detect is 2 picogram per microliter. Yeah, so even with our spec is defined for 5 picogram per microliter, but in some specific way we can detect to very low concentration. And for the cell-free DNA, usually the concentration is not quite high. So this cell-free DNA, the concentration is about 4 picogram, and it can be clear detect from here. Yes. So the last one is the reproducibility, as we just see. Not only for the large fragment size, for the small fragment size, we also can easy to achieve that. And this is also the later we just see. And the red column, that is the 307 base pair, and we run 50 times. 50 times, yeah. And after that, we measure the CV, the sizing CV. That is only 0.8%. So that is quite awesome. Yes, and we also run the large size later. That is the last one is 23 KB, and the CV size is below than 2%. Okay. Yeah, so now before we close today's session, I would like to quickly summarize the key take-home message for you. The first thing, the QSEP platform is a powerful sizing QC tool for third-generation sequencing, whether it's compared to a femtoplots or actual hi-fi sequencing data, the result quite matchable and also very reliable. And secondly, the workflow is very high flexibility. Single sample can can be analyzed. Few sample 1 to 96 sample can analyze, which means that you can run a quickly analyze whenever you need, and even though you have only small amount as well. And thirdly, that the QSEP platform provide very high sensitivity and a high resolution. So for this sensitivity, we can detect down to 5 picogram, and the resolution we can detect below 500 base pair. We can resolve from 1 to 4 base pair. And finally, the QSEP platform is a very broad application. Not only can analyze large fragment DNA, but also can analyze DNA, RNA, and also protein analysis. Yeah, so let's wrap up. Remember our simple message: the good quality and the great data. Okay, so in the end, I really want to share something exciting with you, all of you. The QSEP 100 second version now is officially ready. Yeah, and we want to make we already made several key improvements to enhance both user experience and also system traceability. The first one our major update is we add the built-in LED light inside. So now we have a spotlight on the sample holder here, and that user can easily to put the sample inside and see our tube clearly. And we also enlarge our sample door, so that's easy to handle the sample and comfortable for using. And secondly, now we reduce the minimum sample volume for 0.2 PCR tube from 20 microliter down to 10 microliter. So now user only need to load 10 microliter for 0.2 PCR tube. That is very great news. And for sure, so for this second version of QSEP 100, we still support the microvial. That is 2 microliter. It's acceptable as well. And last one is the customized QSEP 100. Now it supports 342 well plate configuration, makes suitable for very high throughput application. Yeah, and with those upgrades, we want to deliver more intuitive and user experience and modern designs, and also want to increase flexibility and meet different labs' needs. I hope you enjoyed today's sharing, and if you have further questions or need any information, please free to let us know. Thank you.
Thank you, Alice, and thank you all three speakers for these excellent presentations. We now have some time for questions. If you have any, please use the Q&A function and our speakers will be happy to answer. And we hope you enjoyed the webinar and found it helpful for your research.